Going over FRQs. So things that I definitely want to go over today. Any questions you have from Unit 4, I want to go over the Unit 4 FRQs that were in the progress check. And then I have more info on the exam, like what the questions are going to be like. Any questions you have for me first? Any, like, do you want me to go in a certain order? Like, do you want me to go over the exam?
Can you explain what the phosphorylation cascade is? Because I still kind of don't really get that.
100%. Okay, so let me share my screen and let me just get a whiteboard here. So a phosphorylation cascade means that I already had a receptor and I already had a ligand. So something changed inside the cell. So inside the cell, you have these molecules called kinases, and they're regulatory molecules, meaning that they can make reactions happen, but they're normally off. So when they're off, most enzymes are also off, meaning they're in that inactivated state. Oops, I gotta let people in. All right, so meaning that they're not working. And so if they're not working, some of your enzymes don't work until they're activated, because remember, you can have allosteric enzymes, which means there's a certain region that if they're bound to, they don't work. So a kinase would turn them on and make them work. So in order to make them work, you need cyclic AMP, which is a secondary messenger, which is usually due to this G protein breaking off and binding to adenylyl cyclase, and then adenylyl cyclase makes cyclic AMP. The cyclic AMP adds a phosphate group to a kinase. So a phosphorylation cascade means multiple kinases get turned on, sort of like dominoes. You get turned on, you get turned on, you get turned on, you get turned on. And the way they're turned on is by adding a phosphate group to them. So when they have a phosphate group, they are phosphorylated and they are active. And then that activated kinase can go turn on an enzyme, and then an enzyme is going to catalyze a reaction, and that's your response. And the reason why a cascade is important is because it's not a single switch and it's over. Because if all of a sudden, like, let's say you go outside to put out the trash and it's still like somewhat dark out and there's an animal there, and you're like, oh my gosh, it's gonna whatever. Your blood sugar does go up, but it doesn't go up so crazy, because all of a sudden you realize, like, oh, that's the cat. Like, that's not gonna hurt me. That's not something terrible, whatever. So it gives you a chance to, like, slow down your reaction or respond and stop your reaction.
Thank you.
What else? Do you want me to go over exam stuff first?
Yeah, can you?
I feel like people always want to know, like, any exam updates that I get. So I'm going to.
Can I actually ask you a question about exams first?
100%.
Do you think that colleges aren't going to take the credit since it's open book?
They are taking the credit. They've
already
agreed to take it, and they already, it's a done deal.
Okay, awesome. And that applies for every AP, right?
Yeah.
Okay.
Every AP. Okay. I gotta find. Hang on one second. Let's see. Oh, I can't minimize. Okay. Well. Sorry, I'm just having trouble pulling up. Okay. There we go. Okay, can you see my screen? 2020 AP exam. Okay.
Yeah.
So some of these you definitely already know. So Monday, May 18th. Oops, I got people in the waiting room. Ah. Hi guys. Okay, so you already know it's May 18th. You already know it's at 2 p.m. You know that you can use your notes. Ooh, if you haven't already, you should be creating your system of like, how are you going to do tabs on it and get that done so that you get used to like looking through your notebook if there is something that you want to look up. Based on what I'm about to tell you about the 2 questions, there's only 2 questions on the test. I don't think it's going to be really a looking up kind of thing. But in case you want to, I would be making tabs on your thing now. There's going to be 5-minute time periods in order for you to upload things. So you'll do question 1. You'll have 5 minutes to upload anything that you drew, or if you like handwriting better than typing. I don't think there's going to be a graph, but if there was a graph, you'd have time to upload it. Then the second question, and you have, I think, 15 minutes for that question, and then again, another 5-minute upload time period. Okay.
Do you know if you can have it like open on your computer and your phone? So like, cause I'd rather submit like the picture on my phone, but cause my computer,
no, it all has to be submitted through a single portal. So if you're doing it on your laptop, what you'll have to do is take a picture with your phone, send it from your phone to your email, download it from your email, upload it from there. And we practice.
Okay.
I will definitely do like a timed practice thing with you so that everyone's like, okay, can I do it in 5 minutes and how is it? Basically, the way that you've been doing your FRQs is just like how the exam is going to be. They're going to use a different system. Like it's not exactly through AP Classroom because they have a way of like monitoring your computer, but it's going to be similar to that. Okay, so here's the 2 questions that are going to be on there. The first question is 8 to 10 points, and the second question is only 4 points. And the first question, so I imagine the first question is going to have at least an A, B, C, D, because I feel like a lot of them on the AP Classroom right now have A, B, C, D. So it'll be relating to an experiment where they give you data or they give you graphs, like either a table or a graph, and you have to analyze it. And so much like the one that I'm going to go over today, they gave us 2 graphs on one of the ones today, and then they asked you a series of questions about it. That's how question number 1 is going to be, and you'll have 25 minutes to do it. Then question 2 is not about an experiment. It's about like something that's gone wrong. So again, they're probably going to give you data like, hey, if this happened and you realize this, what's going wrong in this scenario? So it's taking what you know about content, and instead of you having something where you can just directly look up the answer, it's like, what did that do? Basically, if you know the content, you can think about what went wrong and write about it quickly. And if you don't know the content, you're going to spend your 15 minutes looking it up. And then by the time you get to your answer, you're just not going to have enough time. So I think that's how they're going to be able to distinguish how much you know. All right, let me go to the next question. Whoops. So the first question, these are what they, like, what we think. I just met with a bunch of AP teachers, so this is what we came up with. So explaining the concept, how did they answer their question based on what method they used to collect their data. Representing and describing data means like that they have a data chart or a graph, and maybe they'll have you, like the next one, the analyzing, maybe they'll have you calculate a rate, or maybe question how good the data is based on maybe error bars. The more error bars there are, the less confident you are in that data. Who just joined us? Karina? And argumentation is you saying, okay, I think that this did prove what they hypothesized in their experiment because this is the data they got. So again, I really think the one that we go over today is just like this, just like this. All right, and then part A, okay, so he really went through like every part, describing the process, providing relevant characteristics. Sorry, I just have to keep letting people in. Vincent.
Vince, happy birthday.
So describing patterns or causes or parts of an experiment. Explain. Again, I'm going to make up a practice one that's like this. Identify.
Could you upload this PowerPoint to like Schoology?
Definitely. And I'm going to do it as a PowerPoint because this is pair something. He uses pair something with this classroom. So that's why I'm skipping past these ones because, oh, pair deck. Pair deck interactive slides is what he uses. But I'm not going to ask you those questions. But I am going to go through, I will put this on my Schoology and I'll come up with sample ones where you have to do exactly these steps. All right. Like justifying. And then the second, oh, no, this is analyzing data. Oh, this is a really common one. Analyzing data is like, what's the relationship between this and this? So like as the temperature goes up, the rate of photosynthesis goes up, something like that, based on a graph. Or as the concentration of substrate increases, the enzyme was not any more active, like it stayed exactly the same. And then calculating, personally, I feel like they're gonna ask you a rate question if they did say have your calculator. So I feel like there will be a small calculation one, and then it probably would be a rate because that's just change in Y over change in X and sort of something that everybody could do, nothing too challenging. Part D is always about like evaluating and seeing if their predictions were met based on the data. Or predicting, like if you did this, what would you expect to happen? Oh, null hypothesis is a good one. So let's just go over what that means. Again, a null hypothesis and an alternative hypothesis. So a null hypothesis is if you do not expect there to be a change. So a great example is like the pill bug lab. If you put red construction paper on one side and green on the other, the null hypothesis is that it would not prefer one side over the other. So you do not expect to see a difference. An alternative hypothesis is they do not detect green, so I think that they'll go to the red side. Something like that is an alternative hypothesis. Usually they give you something like, I think fruit flies like sugar, so it's going to go to the sugar side. Supporting a claim, you've literally done that since high school started. Justify, you've done that. All right, question 2. So all of those were different parts that there could be for question 1. Question 2 is shorter and it's not related to an experiment per se, it's more related to a concept. So explaining a concept. So part A, Provide relevant characteristics of a specific topic. So this is where they're looking more for your content. Describe the factors that influence if a trait will show up in the kids or not. Describe the process that produces oxygen during photosynthesis, like things that are more content driven. Explaining, again, you just writing out your reasoning. So those are always your explanation, your understanding of it. My advice is write down as much as you can, as quickly as you can, because if you just start looking up everything, you'll probably run out of time. Look things up to check. That's what I would say. Check and make sure you're on the right path, and then just write everything you can think of. And then another part of this second question is if there's a disruption or if there's something different. So let's say that the concept is about making proteins. So you know making proteins is based on your DNA. Your DNA has to get transcribed into RNA, and then it has to get translated into a protein. Let's say there's no transcription factor. What's the disruption? What would happen? Well, if there's no transcription factor, there's nothing bound to that TATA box, the promoter section of the DNA, which means RNA polymerase is not going to bind to the promoter, and it's not going to form mRNA. If you don't form mRNA from that gene, you're not going to make the specific protein that you need. An example of this slide right here. And then the last part of the second question, supporting a claim, is rationalizing what happened based on what you predicted. So like, if it was just normal situation, I would predict that the protein for that gene would be made because the promoter had the transcription factor, it did transcription, it made messenger RNA, it spliced out the introns, it left through a nuclear pore, it went to a ribosome, it read the tRNA anticodons, and it built a protein. It put together amino acids with peptide bonds. That's reasoning. That's justifying why it would have made the protein you expected. I think that's the end of that. But again, I will give you several examples of questions that could be like number 1 and like number 2 so that you can practice them all. All right, now I'm going to share a different screen. Can you hear my family? My family is so loud right now. Very loud. Okay. I'm going to share a different screen. I guess I could do that. Yes. Okay. Can you hear me if I use these?
Yeah.
Yes, you can hear me?
Yeah.
Okay. All right.
Hello?
Hi.
Yeah.
I think he's on the phone.
All right, I'm trying to share. Actually, I know what I want to do. I want to go into that app.
Okay. Yeah, I don't really know.
Luke, mute yourself.
Okay, I'm on the Zoom right now, Mom, so I gotta go.
Oh, now it's not letting me upload. Okay. Annoying. All right, can you see the big idea sheet on the screen?
Yeah.
Okay, so any of these that you want me to go over?
Can you go over, I think it's like towards the end, the diagrams I'm still kind of confused on, and I know you said those are like, not those, I get that. Yeah, like these ones.
Okay.
Not that, I got that one, I got the muscle one. I think the page Yeah, can you go over that one?
Mm-hmm. All right. Okay, so you always want to, any of these diagrams, find the first arrow. So the first arrow is right here. And so the first arrow is showing that this hormone is made by the hypothalamus, and that influences another organ, and that makes another hormone, which affects another gland, which makes this chemical. So basically, this is definitely feedback, and the reason why it's feedback is because of these arrows. If you go back and inhibit something else, you are able to control how much the hypothalamus makes TRH and how much the anterior pituitary makes TSH. So that if you make too much of this, so put a big plus sign, you make too much, it will inhibit, it will negative feedback and turn off the anterior pituitary. Then when eventually you don't have as much of this left, you're not going to turn this off anymore, and you're going to start making it again. So that's just really a feedback loop.
Okay.
This other one is showing how your ligand, this one is inactive. And so I know it's inactive because it's not a match. It doesn't fit in this receptor. So in order to fit in the receptor, you have to have an enzyme cleave it or break it or cut it a certain way. And once it's cut, now it's active, which means now it fits in the receptor and it works.
Okay.
All right.
Can you explain, it's like towards the end, it's how is mitosis controlled, and you explain like how to pass the checkpoint.
Oh, yeah. I was so confused on that.
Yes. No.
Next page. Yeah, number 8.
This one?
Go up one. This question.
Okay. So, cyclin. Oops. Okay. Cyclin is a protein. So I'm just going to make this protein right here. This is a cyclin. So cells only make cyclin when they are undergoing a signal transduction pathway where they received a message like, here's my cell, and let's say this is a blood cell. Your blood cells make new cells every 6 weeks. So let's say time is up and this blood cell receives a message, and that message says you need to start making cyclins. So cyclins are the product of a signal transduction pathway. So my cyclin level, if I graphed it, just went from like nothing to way up here. The cyclins bind to Cdks. So this is my CDK, cyclin-dependent kinase. And remember way back in the beginning when Luke asked me about phosphorylation cascade? A phosphorylation cascade is using a phosphate group to turn on a kinase. This is a kinase. So when it has this on it, it's already been phosphorylated, but now it has this on it. So now it is actually on. And here's my cell cycle. So G1, S, G2, and mitosis. These are all the different stages that this cell could be in. Originally, I started off in G1 and my cyclins were really low. In order to pass and move from here to here, my cyclins have to be bound to CDKs, and that level has to be high enough to pass the checkpoint. If I pass the checkpoint, I'm now going to take all of my DNA and replicate it, because that's what S means. S is DNA replication or synthesis of DNA. So once,
how do you know when it's enough?
When it's
like,
enough?
Right, to like pass the checkpoint.
So there's a certain value, it's called a threshold, like there's a certain value that it has to be high enough for the cell to pass. If it's not high enough, it doesn't make helicase. So if you don't make helicase, you're not.
Right, and then you can't do DNA replication. Okay.
Yep. So it's purely based on the value. If you have enough of it, you're going to do that. Think of it like this. Do you sweat all the time? Nope. You only sweat when your body hits a certain temperature, and then you're like, oh my God, my body temperature is too hot. I have to sweat to get some of the heat out. So it's when you reach a certain amount. Or you only make insulin when your blood sugar gets to a certain value. So there's always like a threshold. So there's a threshold of cyclins. Cyclins bound to CDKs have to get measured, and they have to be high enough to actually pass that checkpoint. The role of cyclin is their level goes up and it goes down, because the second part of this is once I pass this checkpoint, break it, break it, break it, break it. Because heaven forbid I make it the whole way around and I never broke these down. I'm just going to pass it automatically. And that's what happens with cancer cells. They start dividing because it passes checkpoints without meaning to. And the reason why is it never degraded the cyclins in the first place. And then a kinase, you always know that this is an activator. You have to activate that to actually do something. So this binds to this, activates whatever process. Hey, now we can pass the G1 checkpoint and we can do S. How about this diagram? Whoops. They love those.
I was like iffy on it. Could you just like run through it?
Yep, for sure. Okay, so we start off with the first arrow. Always find the first arrow in the pathway. So the first arrow is due to DNA damage. So this is a problem. We all know DNA damage is a problem. So let's say it's literally because you were out in the sun. You're out in the sun, too much UV. UV can damage your DNA. So when you damage your DNA, it causes this cascade, a kinase cascade. So that's the same thing as phosphorylation cascade. It means signal transduction pathway. That pathway makes a protein. So this is the protein that it makes. This protein, when it has the P on it, I know it's active because that means it's phosphorylated. So DNA damage causes P53 to be made. P53 binds to your DNA and it causes transcription. So when something binds to your DNA and makes transcription happen, it's a transcription factor. So now,
what happens with the inactive one though?
The inactive one is degraded, which means
okay,
nothing happens to it.
Right.
It doesn't get phosphorylated, it's not working.
Okay.
Okay, so transcription, I know this is what's going to make my messenger RNA, and then my messenger RNA is going to go to my ribosome, and my ribosome is going to make a protein, and there's the protein. So protein was made here, and now protein is made again here. This protein, P21, this P21 protein binds to active CDK complex. So remember how I did a cyclin like this and then the CDK complex? It binds to it and makes it inactive. If it's inactive, that means no mitosis. It means I'm not going to pass my checkpoints. If I don't pass my checkpoints, I'm not going to do mitosis.
And that's just because of the DNA damage, right?
Mm-hmm. Yep. So the question saying like, what effect did DNA damage have and everything else, and it turned off mitosis.
Right. Okay, that makes a lot more sense. Thank you.
Mm-hmm. So this is a question type number 2. So you know how I went through question number 1 is going to be based on an experiment. Question 2 is going to be based on content. This is a question 2. It shows you a good diagram. It relates to a problem. It relates to content that you know and multiple areas of content. You know about signal transduction pathways, you know about transcription translation, and you know about mitosis and cyclins. So this is the kind of thing that I definitely see for the second question. And the great thing is, the thing that like is very relieving to me is that they're not, the old AP would just ask you 2 questions and like, let's say you straight up didn't know one. Oh, well, too bad for you. Now it's like part A, B, C, D. So you have so many chances to like show what you know. And make sure you answer every single part. Even if it's a guess, you're answering it, because you know how many teenagers are going to panic and like leave half of it blank? Tons of them. So don't leave anything blank. At least take your best guess at it. Then you have a chance.
Can you go over the diagrams on the 3rd page?
Mm-hmm. Oh. Let me just make it a little smaller. And the housing moved fine. All right, so this is the first one. So in this diagram, the first thing I notice is this red guy. So that's definitely my ligand. That's my signal molecule. Almost always it's a hormone. So hormones are proteins or steroids. And this one, would it be a protein or a steroid based on that receptor? If the receptor is here on the cell membrane.
I think protein.
Yes, this has
Because it can't go through.
Exactly. If you had a steroid hormone, it would go in here and the receptor would be in the cell, in the cytoplasm. So this is a protein ligand. This is my receptor. So this is step 1, which is reception. Step 2 is transduction, and step 3 is response. This is my phosphorylation cascade. So this is my kinases getting turned on, getting activated, usually by cyclic AMP. And then this is my response. So maybe I make a protein. That's the most common thing. Or maybe I activate an enzyme. Those are 2 really, really common things. One because you turned on DNA, or one because you turned on an enzyme. That diagram okay? Okay, second diagram. Okay, so again, I have step 1, reception, step 2, transduction, step 3, response. So this time, here's my first receptor, here's my first signal. It's not a protein, it's literally light. So light turns on, and you know this works in your body too. Like in order to activate vitamin D, like you need a certain amount of sunlight. So even though you're not a plant, sunlight is still affecting your body. So there's the receptor, there's the light. There's 2 pathways activated, 2 paths. This path is to open a calcium gated channel. So gated channels will let ions in, but only when they're opened. So this got the signal to open the gate, let calcium move from high to low into the cell. Once calcium is in, calcium does some activation over here. And this one, usually when they draw a shape like this, it's a kinase, but I know it's part of kinase anyways because it's transduction. So this is my phosphorylation cascade. And this forms a transcription factor. And the reason why I know that is because it's going here and binding to the DNA. So this is my active transcription factor right there. It's telling RNA polymerase where to go. And then this calcium turned on another path that activated another transcription factor. So you've activated multiple genes, and in the end, when you activate a gene, you're making a protein. This step is transcription. This step is translation. Because transcription made the RNA, and then making the protein from the RNA at the ribosome is translation. Hey, baby. Because I look so good with my gigantic headphones on. All right, next question. What else from this packet? Everybody get the difference between direct contact? So like if 2 cells are touching each other, they could tell from one cell to another cell. Like right now, immune systems are definitely telling other immune cells like, hey, I'm a macrophage. I digested this thing in your body and this is what it looks like. So it comes in direct contact with like helper B cells, and those helper cells are going to be like, okay, I need to make all these chemicals to warn other cells. The chemicals they make go into your blood. So that's not direct contact, that's signaling. So if it's direct cells touching each other, that's direct contact, versus a signal is when it sends out molecules like hormones interleukins, things like that.
Can
Mm-hmm.
you go over the FRQs?
Mm-hmm. I haven't gotten to that yet. I will. Is there anything else on here before I go to the FRQs? Karina, I think you missed too. At the beginning, I went through some stuff about what the 2 questions on the test are going to be, and I will be posting that all on my Schoology.
Okay.
I don't know the content of it, but I know the format of what it's going to be like. Okay. So let me stop sharing that screen and now share the AP. Hang on. Well, I'm bringing this up too. Let me tell you that the only way I was able to post the Zoom was to make it public. Every time I made it private, anytime I shared it, it didn't work. So you do not want me posting it because every once in a while it shows your pictures over on the side. Let me know. I've just shared it with people that asked for the Zoom. Oh no, that's not true. I put the last one on Schoology. All right, I do not want Unit 1. I want Unit 4.
There's like no one on.
What do you mean on this call?
Yeah, there's not a lot of people on. I feel like there's usually more.
There's 16 of you on. But I don't care. Like, to me, these are my like office hours. So like, of course, I'd love it if every person was on, but there are some people that just, I'll do my unit review, I'll submit it. Like I literally am having 100% participation. Like everyone's turning in their work.
Right.
So not everyone.
That's what it is. Yeah.
I don't know. I like these, but.
So do I.
So this was the first question. So the beginning, it's going to be really important that you like take a deep breath, read through what they give you first, because the reading helps give you a frame of reference of what they're about to show you. If you look at the graphs that they show you and you're like, I don't know how to do this, and you panic, forget about it. Like you gotta like take a breath, read it. It will make sense once you do it. So growth hormone and insulin are both protein hormones that regulate carbohydrate metabolism by liver cells through the activation of intracellular signaling pathways. So in my head, I'm going, okay, I have 2 different ligands and they both are the signal molecule for an STP, signal transduction pathway. Researchers investigated whether the 2 hormones, do they trigger the same or different pathways in these liver cells. They added either growth hormone or insulin to cultures of liver cells, and at several time points, they purified the protein that was being made and quantified it. So they measured how much protein was being made. So I look at these graphs and right away I see, all right, here's the level of stat 5, and that's with A. So A makes the stat 5, and or one of them makes the stat 5 and one of them, B, makes the kinase B. Oh, growth hormone makes the A and insulin makes the B. All right, so describe the first interaction that triggers signaling to begin. We just did this. What's the first interaction that triggers signaling to begin? What do you have to have? Not even for this one, for any signal.
The ligand that binds to a receptor.
That's the answer. 1 point for you saying in order to turn on the signal transduction pathway, you need a ligand to bind to a receptor. So that's it. B, identify a dependent variable in the experimental design. Actually, my fan is on now, so.
Negative control for the effect.
You hear me now?
Yeah.
Okay. So the dependent variable is what you measure. Independent variable is what you change. Dependent variable is what you measure. So you measured the level of phosphorylated STAT5 and you measured the level of phosphorylated protein kinase B. That's what you measured. And that's worth 1 point. The second part of the question is identify a negative control for the effective hormone condition.
What does that mean? Negative control.
They've asked this now 2 times in a row. So I feel like, actually, I'm going to put that on my little list. Negative control. So a negative control is the same thing as a baseline. It's what will it make if you do not add the enzyme, the signal molecule, whatever the experiment is adding, don't add it. That's what a negative is.
Okay.
So what would you have if you didn't add growth hormone and you didn't add insulin? What would the level of STAT5 be, and what would the level of phosphorylated protein kinase B be? Like, does it make it without it? That's basically what a negative.
Would it just be 0?
Well, you're assuming 0, but what if there's something else in the cell that makes it, and it's not
Right.
relying on this signal transduction pathway? Maybe it just makes it because it's noon. Like, you have to do every experiment with a negative control. So like, for example, you did your photosynthesis reaction with the DPIP. You did it with no chloroplasts in there. That's a negative control. If you don't have chloroplasts, you can't do photosynthesis. So you're not expecting the blue to change clear. That's a perfect example of a negative control. Another negative control, you did it with chloroplasts and no DPIP. Like, you're just making sure the color isn't changing based on something else, that the color only changes based on the addition of the 2 things. So for this experiment, your negative control is measuring the level of these 2 proteins
without the growth hormone, right? Or the insulin.
No growth hormone, no Cyclin, no insulin. I don't know why I said Cyclin. All right. Then the last one, identify a likely reason why the researchers continued their experiment for 240 minutes. I would say they did it for that long for 2 reasons. Number 1, they want to see what happens to it over time. Like, will it stop working? Like, look at A. It makes it and then it goes way down. Whereas B, it makes it and keeps it up. So like, they want to see how the level changes over a long duration. And then the other reason is maybe you don't know how long it's going to take to start. So like, these both have steep curves at the beginning, meaning that they really started very quickly. They went from 0 to 90 or 100 in just the first few minutes. But you never quite know. Like, was that just the initial spike? And then maybe it's going to be even higher if you wait longer. So those would be the 2 things I would say that would be acceptable answers of why do you wait 240. Next one. Describe the hormone and signaling protein combination that produced the greatest and most prolonged response. B. Totally B, because look at, we've created almost the same response in the beginning, but B stays prolonged. So B made the most over the longest time period. Then the next one says, calculate the rate of decrease. Okay, so let me just hit this button so I can draw on the screen. So for A, from minute, it said minute 30 to minute 10. So that's, all right, so anytime they ask me for a rate, I want change in Y over change in X. So this to this, so 100 minus 35 is 65 divided by 20, 65% over 20 minutes. So that's equal to 3.25% per minute. That's the rate. Yes?
Do you have to do it for both or just the one?
No, it said to do it for the stat 5.
Oh, okay.
If they give us like that thing where it says like upload, how are we gonna know, like, how should we upload that?
Yeah, that's actually true.
You take a picture of it.
Will we just, are they going to be able to read it like through the camera on our computer?
If you do this.
Right, yeah.
When you do this, you'll see it on your screen, and you have to make sure that it's very legible.
What if it's like
Didn't you say you
clear?
just email it though? Take a picture on your phone and just email it to
yeah,
yourself?
you could do that, but
Yeah.
as long as you have enough time to do that, right?
We'll practice it. I do think 5 minutes is a long time, but I know some of you probably have better internet than others. Like,
We're allowed to go to our email during it, right?
But when like 5 people are on Zoom at the same time in my house, it's not good. What was the question, Shay?
We're allowed to like go on our email, right? Like we're allowed to leave like whatever website this is going to be on to get the picture. Because you know how they have like the monitoring of what we go on? Like, are we allowed, like I have Yahoo, am I allowed to go on that? Because technically you could look up answers on that.
I believe so. I guess I'll get more and more information as it gets closer.
Mm-hmm.
But they did say that I was in on a webinar for all AP teachers, and they said that you could take a picture and send it to your email and download it. So I think so. I think that when you get something from an online source, they know the wording, and they have detection software that know how you write versus how something that you cut and pasted looks.
Right, yeah.
In my mind, like anything that you're going to look up, it's not going to be helpful, because these questions, if you Googled any of these questions, you're not going to find the answer. This is all interpreting data.
Mm-hmm.
They're purposely going to make the questions be ones that you can't straight up look up. All right, and then the last question, the researchers claim that the similar regulation of carbohydrate metabolism by liver cells when the cells are treated with growth hormone or insulin results from activation of different signaling pathways. Sorry, I can't read it all. Use the data from the researchers' experiment to support their claim. Well, I would say their claim is definitely true, that using growth hormone stimulated the level of phosphorylated STAT5 as seen from the data. And you could even show data, like in your answer, and that insulin showed the level of phosphorylated kinase B to go up. So 2 different pathways because they activate different proteins. And then the next question, oh yeah, this one. Scientists use cells called fibroblasts to study factors that cause non-dividing cells in G0 to re-enter the cell cycle and undergo mitosis. In a laboratory, fibroblasts typically grow and divide when they're cultured in a growth medium that contains 10% serum, the liquid part of the blood that remains after blood clots. If fibroblasts are cultured for 48 hours in the absence of serum, they stop dividing and enter G0. The scientists divided serum-starved fibroblasts into groups to which they added no serum, 10% serum, or 1% or 0.1% serum. Each of the groups with 0.1% serum was also treated to block the production of only 1 or combinations of 4 proteins, all the little P number things, that are inhibitors of Cdks. The scientists grew the fibroblasts for 48 hours and then measured the amount of DNA replication taking place. Okay, so what I'm going to remind you are some things that you know. Here's G1, S, G2, and mitosis. My goal is right now my cells are in G0. I want them to pass. If they pass the checkpoint, they will do DNA replication. So S is like, this table is actually showing me if they passed G0 or the G1 checkpoint or not. If they're in G0, they're not growing. If you can give them, basically the serum is like, oh, give them this special food. If you can pass, then you'll go into this, which means eventually you're going to divide and go through mitosis. All right, now I got to remind you what this is. All of these, they said, are inhibitors of CDK. If I inhibit CDK, I'm not going to pass the checkpoint. Right? That all seems familiar?
Mm-hmm.
Okay. So I'm now just going to do this so I can read the questions. Based on the data for the groups of cells cultured with 0.1 serums, identify which of the 4 proteins tested does not play a role. Okay, so this one went up. So, so these play roles. All these play roles. Okay, this one right here, P16. P16 doesn't make a difference. Whoops, trying to draw. This one. If you don't add P16 to it, you still get the exact same amount. This is the exact same as this. So that's what tells me that the P16 is the one that doesn't matter. Sorry, I have to clear my drawings in order to move. Then the next one says, based on the data for all the groups, identify the groups most likely to include cells undergoing mitosis. Okay, so out of these, 4, 5, 6, and 7. You agree? 4, 5, 6, and 7 are all doing DNA replication, or at least some amount of it.
Wait, how do you know they're doing DNA replication?
Because this says DNA replication right here. Whoops.
Oh, right. Okay. Gotcha.
Yeah. This is showing me. So this one's not enough because I know that these ones are not working because it said if there's no serum, it doesn't work. So if this one doesn't work, then this one doesn't work.
Right. Okay. Gotcha.
I can't pick number 2 because they said only the ones that were 0.1.
Right.
I'd say all of these.
For this question and even the one above, do you have to explain yourself at all, or can you just write like P16 and then write 3, 4, 5, or 4, 5, 6, 7?
P16, 3, 4, 5, 7. Or no, 4, 5, 6, 7.
4, 5, 6, 7.
Yeah, that's it. If it only says identify, you only have to write what it is. You don't have to give an explanation. Oh, the next one, provide reasoning. That one you have to explain. All right, so the next one says neurons, nerve cells of the central nervous system, brain and spinal cord are in G0 and do not divide in response to an injury. The scientists predict that they can use the data from this experiment to help them treat individuals who suffer from spinal cord injuries. Provide reasoning to support the prediction. Okay, so if I give my nerve cells any of these, they should be able to go out of G0, go into S. And if they can go through S, they're eventually going to be able to go through mitosis. So that's why, that's my reasoning. They did DNA replication. So if I gave them 0.1 serum with each of these different CDK inhibitors, it would work. And then the last one, interactions between cyclins and CDKs control the cell cycle. Explain how the presence or absence of inhibitors may play a role in normal cells becoming cancer cells. So if you have a CDK inhibitor, you're not going to be able to pass your checkpoint. So it's not going to be a cancer cell if it has an inhibitor because you can't keep moving on. If you block all the inhibitors, then it could be a cancer cell because you can keep going through mitosis. Adding 10% serum causes DNA replication, so that would allow your cell to divide. But I think what they're looking for is that you have to have your cyclin bind to your CDK in order to pass a checkpoint. And if you have some inhibitor, an inhibitor is going to stop this, and then you won't be able to go through mitosis. I can also see if they added anything. This. Whoops. The response indicates that the lack of CDK inhibitors might let cyclins and CDKs continue to form. Yep. So if you have an inhibitor, you're blocking from passing into S and eventually mitosis. But if you have an inhibitor, then you're not going to have cancer. If you have an inhibitor, you're not going to go through cancer because you're not blocking your CDKs. When you guys do the free response questions and I grade them, do you get to see that little part? I mean, I didn't do it for this one yet. I went on and I started doing a couple people's, but could you see this or not see this?
I have no idea. I don't know.
Because I can post these too. Like the little, all of them have like literally one-line answers. Like if I go back to here, this says, the response indicates that group 3 does not appear to play a role. Oh, that was the P16. This one, the cells undergoing mitosis are most likely to be found in group 7 or groups 4, 5, 6, and 7. They only give this tiny little thing. Like that's all you needed to get the point.
Right.
Which makes sense when you only have 45 minutes. They're not looking for big explanations. They just want you to identify. What else? Anything else, people?
I think I'm good.
I'm good.
Good?
Yeah.
A signal transduction in my opinion, a signal transduction pathway is gonna come up. And it just it turns everything on. So I feel like it's an easy question to ask. Oh. All right, well, I'm gonna sign off, but definitely message me if you're like struggling or you have other questions during the week. Definitely let me know. I'd be happy to go over things with you.
Thank you.
We have 2 more weeks of regular review-ish stuff. So unit 5 next week, unit 6 the next week. And after that, I'm gonna do all FRQ practice that's gonna be like number 1 and like number 2. And I'm gonna do some stuff on the words. So like if it says identify, what you need to do. If it says explain, what you need to do. If it says provide a justification or a prediction, just practicing those words so that you get the points. All right, sound good?
Yep. Yeah.
Oh, and then I was also talking with this group and they were talking about what they want to do after the AP exam. And what I'm assuming that you want to do is like human body stuff.
Yeah.
Yes, 100%.
Anatomy.
Oh, I forgot we never did that.
Yeah.
Like, is it anatomy or body systems?
Whatever you want.
Body systems.
I'll do
And
whatever.
anatomy. Let's do both.
And then
Let's do it all. Let's do it all.
Yeah.
And I can also do different Zooms. Like if you want to learn about digestion, tune in today to this one. If you want to learn about how the menstrual cycle releases eggs or how whatever, I can do anything. How your nerves work.
Quarantine's pushed back to May 15th now too, so most likely not looking good for us to
What
go
was that
back.
being released today?
I think so. I can send you the, Molly sent it to me. It was some article. Cuomo, New York and other East Coast states extend shutdown of non-essential businesses to May 15th.
Mm-hmm. I love your little, did everyone give Lexi their little pictures so you can have posted where you're going to school? That you don't have decision day?
Yeah.
I love those. Actually, I have to go through it. Like, I saw them on her computer when she was working on them, but I gotta like go on Instagram and like love everyone's.
Yeah, they were cute.
All right.
I should get ready for work.
Bye, Mom.
Well, peace
Bye.
out. Bye.